everyone. Take your seat, please. Uh, Welcome to our last session. Hope you noticed. We had a glass of wine, and this is because we have a special speaker here, Michael Klutstein, please, that will connect yeast to vessels and to stress.
Hi everyone. So I hope you had a glass of wine at lunch. Uh, or despite the kind of down that the first talk gave to alcohol and caffeine. But I'm going to speak today about a project which has been ongoing in the lab for a while, and this is due to our interest in very long-term starvation. So I will just start and ask about archaeological vessels. So there is a lot of ways in which in archaeological vessels we can divine or study what was inside the vessel for archaeological purposes. So archaeologists study this by the location and the context, of course, that the vessel was found, by the ceramics and the composition of the ceramics. This is huge in archaeology. Every time I meet archaeologists, I'm again fascinated by how much this is central. And in the last few years, organic residue analysis and ancient DNA, that's becoming really important. And the point I would like to make today is that one of the newest ways in actually studying the contents of ancient vessels is by doing the microbiome. And we focus not on the kind of ancient microbiome, but on the current microbiome. And the point we are trying to make is that in a similar way to the way that maybe 200 years ago people came and put a date tree somewhere, and then the descendants of that date tree still remain today in that same place. The same is true for yeast, or for bacteria for that matter. So in a previous study we did, we established the novel isolation pipeline, and we studied the known beer vessels for this purpose. So this is just the paper. So this is just the paper that we had back then. And then we isolated 6 yeast strains from 21 beer vessels. 110 controllers gave 2 yeast, and there were probably contamination. So I will show you in a minute. So just this mirror distribution is not random. But it's not all our support, it's the species of yeast that we found that were really interesting. So I'm showing you, these are the 6, and just because this is not the subject of today, but just as an example, we found Udutura, which is a known beer contaminator, and Hypopychia, which is known to be associated with honey wine. And we found it in a vessel from the Persian period that was identified by archaeologists to be honey mead. Okay, so this is, by any means, it's not random. But what we wanted to do is establish that the isolation method is effective and specific, and we examined wine vessels as a study case. And the reason we did that, before that, I want to show you that also in modern beer vessels we can find yeast. And we can actually isolate live yeast from these vessels. So these are kind of old-ish beer bottles, and we kind of isolated the yeast from the stuff at the bottom, and they are still alive and fine. You can see them under the microscope. This is really beautiful. They are Saccharomyces cerevisiae, of course, the predominant yeast in wine. And so the whole story of the wine wine vessel isolation stems from this site, Tel Yavne. So Tel Yavne was discovered about a decade ago, and when it started to be excavated, the area around Tel Yavne turned out to be the biggest wine factory known in the world in ancient times. This is from the Byzantine times, so around 1500 years ago, and the size of it is so big, it's like an ancient Pompeii. You can see an aerial like a picture of the area, and you can see all the stepping stones. There is even one which is for visitors, which is decorated and everything. And if on a regular archaeological site we find 2 million shards of vessels during the lifetime of the excavation, here they found 25 million shards of pottery in this site. I went to the site before it was covered again because they have to build the road on top of it, and I picked dirt from the surface, and it was full of pottery, okay? So this is a perfect site because you have both very distinguishable wine vessels that were used, and I will say something about the wine vessels in a minute, and also control vessels, because next to the furnaces there were vessels that were discarded because they had holes or everything, like it's 4 meters high of discarded vessels, so we could take from that. So it's very convenient and works to our purpose. Just saying that the vessels of this wine were known to be around the ancient world, up to England of today. They were shipped all around, and people called them the Gaza or Ascalon amphoras. The shape of the vessel was very typical, like the Coca-Cola bottle of today. But the source where it is, the factory was unknown until they found Tel Avne. So this is a very, very important site. So we established this pipeline. We both kind of squashed the shards and did qPCR analysis to see if we can find Cerevisiae. This was focused on Saccharomyces cerevisiae, this study, and then we can, in the future, scan hundreds and thousands of vessels using this method. And also, we did our pipeline for isolation, which is to dip it in YPD and wait a week and then isolate yeast only with the selective media, and then we get the live yeast and we can analyze it genomically.
You find tetrads or diploids?
We don't know because we see this after it's already a colony, right? So we don't know what shape it had in the vessel, but bear with me, I will show you something really interesting. Okay, so first these are the qPCR results. I'm showing you just a very, very short snippet of it. So you can see most of them are like either from the controls, model controls, or the site controls. They are negative. Also on the site, most of the wine vessels are negative, but only 4 vessels gave us positive results in the qPCR. And then we took all these vessels and we tried to isolate yeast from them. And we got exactly the same vessels that gave us positive results, and only those. In the qPCR, we managed to isolate these 4 yeast strains, and all of them were Cerevisiae. Yeah? And you can see that in Tel Avne, out of 50 samples, 6% were positive. So this is kind of the percentage where we can actually have information that we can work with in an archaeological site. It's not amazing, but it gives us a tool which we didn't have in the past. Just to be sure of ourselves, we did an organic residue analysis. Tartaric acid is like the hallmark of wine production in a vessel, and you can see that the green vessels where they were suspected of wine had tartaric acid and the controls didn't have, so it works like this. The last thing I want to show you is that we asked ourselves, why did we manage to isolate yeast from those vessels and not from other ones which were also identified as wine vessels? And one of the ideas was that the pore size inside the ceramic is bigger in the ones where we could isolate yeast from, and therefore yeast can go inside the pores and hide there until the conditions become better, and then they can be rescued or live longer. So we measured the pore size, and you can see here the positive and the negative. There's a very big difference in the pore size, and in the positive charts we can even see very nice images of what we think looks like yeast cells. So not necessarily they are tetrads. I think that the yeast cells, and this is another paper that we did, can survive not as spores, but as resilient cells, mitotic cells, for a very, very long time, much longer than we thought before. Okay, so the conclusion. So the methods are specific. and the microbiome can be scanned by qPCR, and we found 4 different vessels, and we are analyzing the genome of these strains now. So a few more things. We're almost ready to tell all the world about this, but there's still a bit of work to do. We are recreating wine with it. So there's the Center for the Study of Ancient Wine and Grapes in Ariel University, headed by Professor Eliashiv Drori. So we are working with them. They are the experts. So we are remaking wine. And also we're working on the phylogenetics of this yeast with the biggest expert. There's still one big expert on yeast phylogenetics. He sits in Perugia in Italy, Gianluigi Cardinali. And we're working with him together to isolate these yeast. He finds this fascinating because they are very special in their phylogenetics. They are not like the normal yeast that you find everywhere. So with this I will finish. This is a long-term collaboration with my close colleague Ronen Khazan at our institute, who is a bacteriologist. So we exchange ideas between bacteria and yeast research. It's very fruitful, and is spearheaded by Eden Rosenberg, who is here in the audience. And that's it. I will happy to take questions.
Are they tolerant to higher alcohol concentration?
So yeah, so all this again was shown both in the 2 years surviving yeast in a beer bottle paper and in the beer. The same, and we're working to show the same here. So they are tolerant to high stresses. Yes, more than
To alcohol, higher alcohol concentration.
we haven't checked that yet, but it's on the list. Yeah. Yes.
As far as I know, cultures that they've taken from the wild forms quite unusual colonies until they become domestication to the lab. Do they have some special appearance on the other place?
Where is Eden? Eden can answer that the best.
No?
Not really, but they do.
Like pseudo hi-fi,
Sometimes, but they look shiny and round. No,
They
So
look
they are already domestic.
already
Yes, but
domestic.
do
Yeah.
they are not. They,
in your papers you have a special media to see them, right? It's like a soft
No, for some of them, but not for all.
But maybe you need to go to this like soft
I have seen some phenotypes, of what you have in your papers, that they have these little coming out of the colony, invagination of the colony, but it's not very common, I must say. Yes.
The yeast that you managed to revive as colonies, were they spores or stationary cells?
Exactly, so that's the question that was asked by Shai before, and I think we don't have a definite proof, but we do know that they can survive as just mitotic vegetative cells for a very long time.
If you do RNA-seq, you would be able to tell the difference.
No, because we don't have the original cell, right? The original cell was already grown.
You can sequence the original, at least the major population.
We tried different ways of going back to the vessel and looking at nucleic acids. Okay, but not for this reason. We wanted to do ancient DNA. And the problem is, this is not like any other study where you work in the desert and find a little bit of ancient DNA. This is a very rich environment in DNA. So everything here, if we will extract RNA from the vessel, and you can't do that, everything will be contaminated. So it's a problem.
But you can, if you have sequence, you can know which sequence it belongs
We can
to.
try that.
And
I'm
you
not
can
sure I
tell-
trust
and you
this.
can tell
Yeah.
whether the majority of the yeast RNA came from stationary cells that maybe died or even stay alive, or from spores.
Yeah.
Because the repertoire of RNA is completely different.
I agree with the notion. The question is, it really interests us as yeast researchers, but I'm not sure that it really matters in the way of the mechanism, because it's thousands of years. So right, so they would be in a spore and then germinate and then go back to being a spore and then germinate and stay as mitotic cells. They can go back and forth, I think. Okay, thank you.
With that, so now we go on to our de-stress panel. So I would like to ask all of our panelists to come and join us here on the
Moti,
stage.
Leah, Ella, and Tomer, please have a seat.
And the idea behind this panel is that we just heard from Liat that the thing that keeps us most stressed is uncertainty about the future. So there's no bigger uncertainty about the future than for people in academia, and especially students and postdocs in the audience, where it's very, very hard to know what you want to do next, how will you do it, will it work out, many things that you're all dealing with. So we brought...
Can we turn off this projector?
No, it's we're stressed.
We like just to see them.
Video, exactly. So we brought the most relaxing...
See them suffering.
And it would be great if we could start off, we will start off with them introducing themselves and maybe giving us one tip on how you manage to remain unstressed despite the stressful academic environment. And then we'll open the floor for questions from all of you. So please think of whatever you want to ask. It can be about career trajectory, it could be about life in academia, work-life balance, life choices, anything that interests you. It will be great to hear from you. So let's start from you, Tomer. Ladies first. Just maybe 2 minutes, say your name, where you're from, 2 minutes about your academic track, and what you've been through, the challenges that you've been through, and a tip on how to stay sane in academia.
Okay, so I was preparing to the previous talk and had 3 glasses of wine. It's also distressing for me, and so bear with me if I'm saying shtuyot. My name is Leah Geber. I'm from the Ben-Gurion University. I started as a chemist, and in my postdoc I actually switched to genetics and molecular biology. This was in Johns Hopkins, when I was introduced to the beautiful world of cytoskeleton and motor proteins. You heard the student from my lab, Mayan Sadan. And after my postdoc, so first of all, I'm representing what is called two-body problem, in the sense that my spouse is also a scientist. So we need to find places for 2 people in every step of our career. So this was a problem in the postdoc. I sent, it was back a few years ago, and so I sent 35 application letters by mail, like regular mail, to find a postdoc. My spouse did the same, and we found a few labs, and same was coming back. We ended up in Ben-Gurion University, where I started my lab on chemistry and motor protein, chemistry, biophysics, molecular biology, and genetics. And I'm trying to educate, one of my goals is to educate the new sciences generation in this interdisciplinary approach to science, which I love very much.
No, the next round will be the tips.
So hi, I'm Ella Pig. I'm from the University of Haifa at Oranim. I did my master degree at Tel Aviv University, then my PhD at the Technion with Danny Kassel, and then I really didn't know what next. And I decided, it became that Michael Glickman looked for a postdoc for a short term, and I took it, but then I found you can say, golden position as a chief scientist in a startup company, worked on preeclampsia, which is רעלת הריון, and it was not a very good time for me. I mean, the salaries were very, very high, but I felt like a bit prostitute, Since every month I said, this is the last month, I'll just buy this and that and that, and then I'll quit. I didn't like it since we always looked for the money, for the next money, and not for the biological question. And every time when I found something interesting, it became that I have to leave it, since it's not the interest of the company. Then I went to Michael back, עם הזנב בין הרגליים, and I told him, listen, I need help. I need 1 more year with you in order to find a postdoc outside of Israel, since I want to go back to the academy. And that's actually what I did. I returned to him for 1 more year. Then I went to Yale University, and that was the most interesting part for me, because the most interesting question I always asked was the connection between environment and intracellular signaling. I went to a specific lab that could give me these tools, and that's why I got back to Oranim, because in Oranim most of the researchers deal with environment, and I thought that it will be a great opportunity for me to study molecular biology and environment together, and it took me 19 years, and that's what I'm doing now.
Hi, my name is Moti Choder from the Technion. I did my PhD at the Weizmann Institute. I did my PhD at the Weizmann Institute, and then moved to Boston area, did my postdoc, and then I needed to decide what I'm going to do when I'm grown up. And there was one thing that I knew for sure, and this is that I'm not going to study animals. Animals are my best friends, and I'm not studying friends. So the only real option that was left for me was yeast, and yeast genetics, and yeast molecular biology. And I think that our science, the science that we do, is the reflection of our personality. Namely, I think that no matter what I would have started with, I would have ended up doing more or less the same thing. I asked myself this several times. So if we are left with enough time and relatively enough freedom, eventually it's like art. Eventually you are doing what your personality is. And what interests me all the time, all along, was communication. All sorts of communication. So I think that if I had started working on cancer, which I wouldn't because this involves animals, but if I had done it, I would end up working on communication among cells within the body, among cancer and non-cancer, exosomes, all sorts of communication, because this is what interested me. And right now, what we are doing is to study the communication between various processes within the cell. Now, I've been through a lot of stress during my career. Some of you know, and I'm not going to elaborate on it because it may take a whole day. We just say that if you are passionate about what you are doing and you're persistent enough, you can achieve everything.
Hi, Tomer Raviv from the Hebrew University, this campus. So actually I started my research careers with animals. I was doing studying lipids in shrimps, marine biology, and decided to continue in the Shiva Hospital for PhD in the lipid department, and then moved to Yale when I met Ella. Actually, I moved first to California, from animals to tissue culture research, EGF receptor, mammalian cells, and eventually end up at Yale with Yeast genetics. So trained both on the biochemical side and also gained a lot of genetic knowledge. And since then, I'm very happy to stay with Yeast. I think maybe all careers, I mean, I cannot think about going through this path without any stress. It's almost impossible. I think the most stressful times for me were actually during my PhD, where I was working on cholesterol metabolism, cholesterol regulation, and I think we have a good hypothesis that actually we have the main enzyme of the pathway is rapidly degraded by the ubiquitin proteasome system. Then it was all in the early days, and we just need to prove it. And we did westerns and all this, looking for ubiquitination. And actually, it took me 3 years of my PhD for one gel. Every day, almost, I would do the gel, change everything, and it doesn't work. A month before I had to finish my PhD class, so 3 months later, I could finish with all the data. So if I will be the first one to give some idea of what, advice for students, so maybe it will be that you need to be consistent, to believe in what you are doing. And I told my students all the time, we're not doing 100-meter run, we are in a marathon. Sometimes you feel bad, find the time you feel better, but if you want to finish, if you have a goal, you can achieve it. Just
Long distances.
long distances, yeah, for sure.
And so maybe we cycle back for the tip. One quick tip on how to survive and thrive in academia.
Well, I think that I already mentioned one important tip, and this is depending on you. If you are passionate about what you are doing, this is the major driving force that we have. And I would say that during a time of stress, what I would do is to go to other fields of science. For example, physics. I love physics, and I listen to many podcasts, etc. Other type of science. I remember that during the first year of my postdoc, I was really depressed because it didn't interest me what I was doing. It was the first and the last time in my life that I did something that somebody else told me what to do. And then I found comfort in reading other types of science, because you need this, I need this intellectual stimulation in order to keep alive. So this is one tip. Another tip, which I'm not sure it's a good tip, but this is what I give you, is to do what you love and not what you think is going to be popular in 5 years. And as I said, I'm not sure that it's a good advice, because I know if you plan in advance and you know that the AI is going to be a major issue in biology in 3 years, and now you come with this equipment, with this tool, maybe you have the upper hand over others. But I would choose to select topics that you love, because if you love, it's like your baby. If you love your children, you nourish them, and you put all your efforts, and they grow up, and become a wonderful individual. So this is the major tip that I would give. Maybe during this session there will come others.
Next cycle. Yeah, so I think I'll give you let's say like this. For me, science is like a playground. And if you feel stressed, just think about it like a playground. You have all the tools around you, and you can just choose whatever, and always you'll fall down and sometimes nothing will work, and then everything will be great. And it's not like every gel is a result that you'll publish in Nature, right? So if you think about it as a playground and read, do a lot of reading, think by yourself. Don't wait to your PI to tell you what to do. Think about it before. Think about all the controls and just enjoy it as a child in a playground. It's going to be fabulous.
Okay, I actually have two tips. For the first tip, I will tell you a story, my personal story, one of them. And the story goes like that. At some point, we discovered that the protein that we were interested in becomes phosphorylated in a certain stage of cell cycle in late anaphase. And then we made a big issue out of it, I got grants, etc. But how the discovery was made, and this is what I'm going to tell you now. So we played with the protein in Western blot, and I arrested cells in different points of the cell cycle by using specific cell cycle mutants that were very prevalent then. And I loaded it on a large gel, large being larger than what we use today, gel of that size. And I wanted to stop it after 1 hour or 1.5 hour run. And then some other person, some postdoc in the lab, it was in Johns Hopkins in Baltimore, had a birthday that day and said, let's go for a drink. And I said, okay, I look at the gel, said, well, it's large and my protein is large, so I can go to drink. And I come back 6 hours later, after a drink, and I see nothing on the gel. It all ran out, the dye went down, and there was nothing there. So I'm thinking, what am I going to do with this? I'm going to throw it in the trash can. I said, no, let's assemble a Western blot sandwich and put it under a car and then see tomorrow. And this is what I did. And this was the first time I discovered that our protein had a gel shift due to phosphorylation. And I was not able to see this result unless I went for a beer and came 6 hours later on. So the first advice, be open, tuned to opportunities. If they come to you, don't throw them away. So this is the first one. Of course, you cannot know. You have to be lucky at some point. But if you do see something, don't throw it away. Just let the luck come to you. Be open to opportunities. So this is the first advice. And the second advice is a very short one, and you probably all heard this. So what is the difference between winner and a loser? Right? Winner is a loser who doesn't quit. To me, one of the most predictive attributes to being a successful scientist is not how smart you are and how well you remember your details, because there are so many smart and talented people who end up nowhere. But just be persistent, believe in yourself, do it over and over again if you can. And if you love it, if you're open to opportunities, if you do it decent control, right, it will probably, at some point, hopefully in this lifetime, will pay off. So this is my 2 tips.
If I can say just something related to your talk, so I heard a talk with Paul Nels, who discovered the cycling kinases, yeah. So his story is he was screening for in Yeast plate for cycling. I don't remember what was the assay.
It was a cycling.
And
the sick one.
Yeah, so he just checked 1 plate, 2 plates. Decided to, after the 40 or 50th plate,
70
to go home. All right. He went home, but he couldn't sleep. Went back to the lab, go to the garbage, pick up the plate from the garbage, and found the first cycling. So never quit. And
so we heard the
look for
same
opportunity.
talk in
No, but my story is true. It's actually
He got the Nobel Prize.
right. Okay.
When the young generation, you have a question to raise. Please do. Yes.
I have a general question, and Leah, don't get mad at me.
חכי בבית.
If someone is passionate about research and learning and like working with the hand and really doing the research itself, should we go in postdoc outside of the country to continue in academia, or are there other options to continue in this field and not just deal with bureaucracy and grants and meetings?
Yeah.
Just wanted to do the research, yes, the research.
Without boring.
Well, you're asking a scientist.
What do you want?
Maybe I can pitch, for the Weizmann Institute, a little pitch. So in the Weizmann Institute, we have a staff scientist position. So every lab has a PI and a staff scientist. So there are about as many positions for PIs as staff scientists. And this is an academic position, it's tenure track. It's for people that have finished a postdoc, not necessarily abroad. It can also be in Israel. It had a successful postdoc and published well. And it's an amazing position where this is exactly what you define. If you want to stay connected to the science, doing the science, being in the lab, not doing the administrative part of the science, then this is absolutely amazing position, and I highly recommend it.
But in the long run, it may be frustrating because at the end, there's someone else heading the lab. It's a trade-off, which we all need to
It
consider.
depends on the head of the
Of
lab.
course, but it's a trade-off one need to consider. And I'm not sure if I had to make the choice today, I wouldn't make the same choice. I mean, it's very dynamic.
I'll just say that I heard once from Yuval Schwan from the Technion. He said to be a staff scientist, you need to have a very big head, very big ambition, to have lots of ambitions, but very small ego. Otherwise, it doesn't work.
It's typical to the brother.
There was also
he's a
another
great, I love it.
question that was raised. Did you ever consider going to the industry? Did it cross your mind?
So I said, yeah. So I worked in a company, and at the beginning it was great. I felt like I'm a chief scientist. I went to conferences, to collaborations abroad. It was the year where the Human Genome Project was just finished, and we could learn about the genes that we were interested in. We purified proteins from placenta, human placenta, and every gene was a whole world. And then in some point, I felt that it's, as I said before, it's useless. I mean, I'm just doing whatever brings money. And it's not so bad, right, to bring money. But if you are a person that likes to think and not dedicated to have, to find something in the next איך קוראים לזה שמוצאים חברה החוצה? Exit, exactly. The next exit, I forgot for a moment. Then I suggest you to go on with academia and think about it later. And I want to say something, another thing about it, about company. I think that the best for me, and I really suggest it to everyone, is to go abroad. If you can, try to go abroad for a postdoc. It's good for the family. It's good for yourself to see the world, to have another position, another perspective to look at life, and then come back to Israel and find yourself. It could be in academia, it could be in hospitals, it could be as a teacher, it could be in companies, whatever. But first, try to go all the way. It's so good.
Okay, yeah. Okay, so last question.
I don't know if it's too big, but when do you feel like you're starting to develop this creativity in science? Because for my master's degree, it felt like driving lessons, and now a PhD, just learning and learning and learning, and sometimes you learn so much, but you wonder when this creativity of, I don't know, PI coming from, and maybe it should be now also, I don't know.
Very good question.
Yeah, I have an answer to it, Richard. I'm pretty sure about it. I've been following a student of mine, and I came to the conclusion that a turning point for any individual is when it starts to have a self-confidence and to trust his or her common sense. Because science is nothing but common sense. Knowledge is important, everything else is important, but at the end of the day, it's common sense. And usually people don't trust their common sense because it's, oh, there are so smart people around. But my experience is that we are not different from each other in terms of our intellectual capacity. The main difference is emotional difference between who will end up eventually being independent scientists or finding him or herself in other avenue of life. So you need to learn to trust your own judgment and realize that your judgment is as good as everybody else. And that's the turning.
Especially your PI.
I just want to echo it with one word. I think that when I look at my career, the best time for me for research was when the PI wasn't around. Okay, so
I say this all
you
the
can
time.
take the risk, and you know the best way to do that. So.
Maybe I can add to Tomer and Mati and say you should practice it. As much as you practice those skills, you know better.
Maybe mentoring juniors can also help in this case. When you have a new master student, you talk and you understand what you don't understand. And they understand.
Teaching is a great, it's a
Absolutely.
great learning. And for you, Shubham, this was also a turning point when you started to teach others.
Yes, absolutely.
No, I saw it.
This is the turning.
I want to add something else because this is a topic about stress. And I want to share with you my recent experience of coping with stress, which helped me a lot, and maybe at least one of them, it will be helpful. It's worth saying. I heard a podcast about Buddhism. And the Buddhism, they have various principles. And there were two principles that I adopted, and it helped me a lot in coping with stress. One, live the present. Don't think about the past and don't think about the future. I'm not saying that don't plan your life, no. But
Don't stress about
emotionally, don't put all your feeling neither in the past nor in the future, because these are the source of suffering. Look at animals. Animals suffer much less. They don't have ulcer, partly because
they live in the
present. This is one principle that helped me a lot. Another, so whenever I'm stressed about, what is going to happen? I'm not going to get a grant. What shall I do? I said, stop it. Don't think of it. You cannot do anything about it. Think about now. So this is one principle. And the other principle that helps more elderly people than most of you here is the understanding that everything changes. All the time, every day, you wake up a different person. You become older, your preferences are different, your friends react different to you, and everything changes. So I accept it as
Be flexible.
no, I accept it in a religious manner. I'm not religious, I'm atheist. But I accept it as a force of nature. This is nature, and I'm in nature. So I should accept that I'm growing older, that my hair become white, my belly starts to develop, etc. So these two principles help me a lot, and I think that now I'm not a stressful person.
Thank you for sharing. one last comment, and then Maya would be back on the time because, okay.
Less than 20 seconds.
Right, so, yes, so I just wanted to add one comment, especially for the young women in the audience. And that is, take risks. Don't be afraid. Because as we heard in the talk, we humans, and especially women, tend to overthink the past, the future. Don't let these things stop you. Because we as women, the imposter type of syndrome, the insecurities, all that, and what is going to be with my family, with my children, what, push all this apart, breathe 3 times, and plan your postdoc. Plan your postdoc is the icing on the cake. I totally agree. No matter what you do later, teaching, postdoc is very important because then you get to live everything that you suffered for when you were a PhD student, right? And only then you can decide if you are creative enough, if you have the guts, if you have the killer instinct. Only in postdoc, in a new environment, not here in our little puddle. Women, take initiative, take risks. Don't think too much about the future. It will be okay. As long as you will stay healthy, it will be okay. So this is
Panel members, and go on with our
אתה לא יכול לשבת על זה.
שאת מעניינת, זה לא אומר שאת צריכה
טוב, so our next speaker is Amina Aroni from Ben Gurion University, and then you have to stay at the end of the session so that they introduce you.
I talk Britain. Okay, thank you for the invitation. After this nice reflection from all the experience. So I will talk about live monitoring of application for progression to damaged DNA. And our lab is interested in eukaryotic DNA replication. Of course, we use yeast as a model organism. And what we are specifically interested is in how replication will progress through different obstacles in the DNA. So we have a number of projects. We have one big project that involves replication through G4 structure, through tandem repeats, mainly triplet tandem repeats that are associated with neurological diseases in humans. We're also looking at replication through site-specific DNA damage. I will talk today more about this system. And we have a big project about transcription replication conflicts. Whoever is interested in each one of these, I'll be, of course, open to more discussion. Now, to connect a little bit to the theme of this conference, it's well known that all of these obstacles can lead to replication stress and genome instability that eventually can lead to cell death or complete stop in cell cycle progression. So this is the main topic of today in this short talk. And actually, the main methodology that our lab uses is actually an approach that we developed a number of years ago that allows us to measure replication in real time using inline yeast cells. So in contrast to a more traditional analysis of replication that is done either in an indirect way or through sequencing at different stages of the cell cycle where you need to, the researchers need to extract the DNA. Here we monitor in real time the progression of single replisomes at a specific locus in individual yeast cells. So actually all these properties, mainly our ability to look at single cell analysis and to look in real time at high time resolution, allows us to really look at a very mechanistic way and to derive quantitative different parameters of the system that we investigate. So how does this work? How does this approach work? So what we use is a well-known fluorescent repressor operator system. And what we do is we integrate into the yeast genome. Now we developed a very flexible approach based on the Cas9 that we can integrate a large operator arrays of several kilobases in a flexible manner in any genomic location. And this operator array here, the LAC operator array, is bound by LAC repressor fused to a fluorescent protein, and this is of course flexible. So we use fusion to hollow protein and then label the protein with silicon rhodamine to have a fluorescent at the far red region. We use a second array that is a TET operator array that is labeled by the TET repressor, of course, fused to most, in most common system to the TD tomato. Now these arrays, of course, were known for many, many years, developed, I think, about 30 or more years ago. So how do we actually exploit them to measure replication? So our idea was to integrate one array relatively close to a strong origin of replication, the other array downstream of the first array, usually the distance is about 20 kilobases. And when we tune carefully the expression level of these repressors, we can generate a situation where we look at the yeast strain, we see two fluorescent dots that actually mark the DNA at the specific location, of course, that the arrays are located. So what we do in the lab, we exploit it to measure replication, is we synchronize the cells in G1, namely at the phase before replication. Then we take them to the microscope. We initiate replication. The first array is replicated, and this, we monitor the intensity of the dot by live cell microscopy, and then what we expect to measure is roughly doubling of the fluorescent intensity signal. Of course, the two sister chromatids are held together by a cohesion that we actually see the two copies as one fluorescent dot that increases in intensity. Now replication is of course progressive. This is just a snapshot. And the second array is duplicated, and then we measure independently the fluorescent intensity of the second dot, and we can measure the increase. And then by doing a very, very simple math, we can measure replication rate. But the most important parameter that we actually measure is what we call replication time. This is delta T. And delta T is actually defined as the time it takes for the replisome to move from the first array to the second array. We look at the midpoint here, so we estimate that this is the roughly, let's say, the mid-array location. So now imagine that we have an obstacle here. Again, it can be secondary structure, transcribing DNA damage. Then we expect that replication time will be longer because the replication will either slow down or stall for a few minutes at the block that we integrate into this location. So this actually allows us to quantitatively measure here in this simple system replication slowdown or replication stalling at a specific block that we, of course, determine. Okay, so here you can see data from a single cell. By now, I think we measure thousands or tens of thousands of individual cells. And you can see here we use the GFP label for the LAC array. You can see different snapshots by progressing through the cell cycle and the increase of the two fluorescent dots, and then the calculation another very important parameter that we can derive here is also the, let's say, an approximation of the G2 phase by looking at the time from replication of the second array till the anaphase that is very, very clear in our system. So we clearly see at a specific time point that the two, of course, copy of the DNA separate. One goes to the daughter cell, as you can see here, and because we monitor only the intensity at the mother cell, we can see this drop in intensity, and therefore we can actually look again at the single cell level and correlate even between slowdown in replication here and elongation of G2. So for example, we have a project by Zohar, who is here, I can't see him now because of the projector, but anyway, that he monitors replication through tandem repeats. I will not talk about tandem repeats too much, but we saw that replication slowdown is associated with elongation of G2, and then through mutational analysis, we saw very nicely that it is actually mismatches that are left behind the fork, and that they are repaired by the mismatch repair system during the G2 phase. So, and a development on this system that we implemented a number of years ago was by Amit Cohen, who is here in the audience, that he was brave enough to take upon himself quite a challenging project. The concept is quite simple. So the idea that we had then is to introduce site-specific damage during G1, namely before release of the cells into S phase, and then release the cells and monitor for progression at the single cell level, how replication progresses through damaged DNA. Of course, this is nice on paper, but in reality it was much more complicated to divide the system. And in reality, what we have done is actually to target Cas9 to a region between the array. We actually developed a third array for this project that we termed a synthetic CRISPR array that contains gRNA target sequences with the idea in mind that if we increase the number of sequences here in blue, then we can inflict more damage at a specific, let's say, time duration and monitor replication through different loads of damage and monitor their response. So this, of course, also took an effort. So Amit succeeded to induce Cas9. Here, I mean, what we forgot to mention, that with Amit, we focused on introducing our basic sites. So for introducing our basic sites using the Cas9 system, we use a dead Cas9 that, of course, is completely inactive, fused to UDG, a mutant of UDG, which is a DNA glycosylase that generates our basic sites only at T bases. So Amit devised a very efficient induction system of Cas9 based on estradiol and the GAL promoter. Again, if anyone is interested, it works beautifully. And the advantage of this induction system, I just said one sentence, is that instead of using galactose, getting rid of glucose, changing media, and so on, we can simply add very low concentration of estradiol, and the expression is very, very high. Another feature, important feature of this system is how to degrade Cas9 efficiently during G1. So we adapted the auxin-induced degradation system by fusing Cas9 to Degron and then adding auxin, and by adding, actually Amit improved it, by adding 2 Degron sequences, we can even add auxin in the presence of estradiol, the inducer, and the auxin effect was much stronger, allowing us to get rid completely of the Cas9 during G1. Then after all these, let's say, after all these protocol of yeast, then finally we release the cells into S phase to replicate through the damaged DNA. Now, I think a final, let's say, innovation or improvement that we had in this project is that we didn't pick, let's say, this silicon rhodamine and TD tomato by chance. We've done it on purpose to release the GFP, allowing us to fuse any protein of interest to GFP to have a three-color system. And here it really made a huge impact on this project that we fused GFP to either RPA. RPA, from the one that don't know, RPA is a single-stranded binding protein in yeast, this single-stranded binding protein in yeast, and it's of course marks single-strand DNA gaps that are formed during replication through damaged DNA. And also we adapted a labeling approach, relatively recent labeling approach for RAD51 that allows us to look at homology recombination events that happens during replication bypass through the damage. So this is the basic system. And again, as I highlighted before, we expected that without DNA damage in kind of control cells, we will see fast replication, and fast replication will be, of course, measured by a shorter replication time, the delta T. And if we have a slowdown due to replication through the damaged DNA, then we have a longer delta T. So this difference, of course, between the two allows us to estimate exactly how long does the replizome either slow down or stall at the damage site. So the nice thing about working really hard on developing system is that if they are wide enough, you can do many things with these systems. So because we use Cas9, we can build upon a lot of work that was done with Cas9 and inflict different types of DNA damage. So of course we can introduce double-strand breaks. It works beautifully in this system with very, very high efficiency. But double-strand break, I mean, we can measure the dynamics of repair of double-strand break, but this is a very, very, let's say, studied subject already. Only 2 minutes? Okay, sorry. So I'll just go through the system quickly. Okay, and we can do NICase. So NICase to introduce SNCs either on the lagging or leading strand. Amit is working on our basic side, and we have another project that we work on mismatches. So what shall I tell you in 2 minutes? So I can't get really much into this. Okay, I just go to some results to show you how it looks in reality. So we saw that if we introduce more damage into the system, then we see more stalling. This was nice because this is again in very high accordance with the literature. And again, what I want to show you here is that we can look at co-localized RPA foci that mark, again, single-stranded DNA, and then we can monitor the dynamics of single-strand DNA gap formation during damage bypass. And this is, here you can see a very nice analysis that we've done in the lab, that it's a little bit complicated, but what you see here is actually the time axis, and you can see how replizome progresses through the damage. The damage is located here, and we saw that actually RPA in some cell population, RPA goes very, let's say, dramatically increases its intensity, and then resolves during the cell cycle. I'll just conclude here, because of time constraint, that after a lot of single cell analysis, mutational analysis, again, I want to emphasize that there are like hundreds of papers on DNA damage bypass, we concluded or could really see how yeast cells that looks homogeneous and they all, let's say, suffer from the same extent of damage, how they can choose between different bypass pathway. One pathway that we identified very clearly is yeast using translesion polymerases that are characterized by the absence of RPA foci, milder replication slowdown, and higher propensity to complete the cell cycle. We term it high viability. And the other bypass that is very famous by template switching, homologous recombination, that is characterized by RPA foci with much, let's say, more strong slowdown. And one of the nice things that we actually identified, this is the final conclusion, is that actually forestalling a damaged DNA is not depending only on the damage itself, but how cells respond to damage. So I really kind of connected to the kind of what we talk on how we are as humans, let's say, deal with stress. So the stress can be the same. Imagine that these are we are all yeast populations. Some can choose a cellular pathway that will, of course, lead to long forestalling, and some will go through it very quickly. So this is, thank you.
Jump over the stress and some try to fix it.
Exactly, yes.
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 Good.
Sorry that I didn't plan the time well, but...
No, but you got the take message.
It's good.
Yes, please, one question.
Hi, so what's the difference in the stalling you observed for the different DNA damage types?
Okay, that's very good. So I didn't show it, but it's a couple of minutes. So it's about 3-4 minutes of difference, quite significant.
Sorry, we have to move on because
sure.
of the time. Thank you so
Yeah.
much. And our next speaker is Reut Nor-Fazan from Ofi Pines Lab.
I think you should do something for wake up like
Yeah.
yes.
Wake up, everybody. Let's get up.
I'm too tired.
Let's go wake up. Yalla. Cool, that's it. I'm not checking. We did checking already. We did something very good. So let's רגע, בוא.
I love watching everyone is taking a seat. So, can you hear me okay without the noise? Okay. So, hi everyone, I am Reut. I'm a research fellow at the Ofi Pines Lab. And he couldn't make it, he's in Australia working. And yes, working. And today I want to tell you about our project, which is a collaboration between the Pines, the Saada Reich, and the Regev Rutsky's labs. And I'm very lucky that a talented PhD student, Bori, which is here, and an amazing master student, Abigail, which is she's now writing, they joined me for this amazing project. So let me start by asking you a question. If I have told you that there is another mitochondrial derived factory to produce ATP, would you believe me?
No.
No.
No. Okay, it's impossible.
No way.
Okay. Martin, you know the answer, so don't. Okay. So let's start. Yeah. So as we all know, eukaryotic cells are defined by the existence of subcellular compartments and organelles which allow localization of the chemical pathways into separate organelles. Now mitochondria are one such example, and it's our main interest in the lab. And as we all know, mitochondria are very complex organelles housing hundreds of biochemical reactions, such as the TCA cycle, iron sulfur cluster synthesis, and ATP production. And these reactions involve substrates and products that flow between the organelles within the cell. and there is an increasing evidence that direct interorganellar contacts are required for this. So elemental iron uptake, for example, has been shown to require kiss and run contacts between endosome and mitochondria. Moreover, based on various studies, mainly from the Schuldiner's lab, it is well known that lipids and calcium are transferred between ER and mitochondria through contact sites. And studies in yeast revealed also contact sites between mitochondria and all of these organs. But the most interesting phenomenon regarding organelle crosstalk for us was the evidence for mitochondrial-derived vesicles, a process inherited from bacteria. And this brought together our lab and the Regev Rutsky and the Saad Reich labs for an exciting collaboration. So let's look on these mitochondrial-derived vesicles, or shortly MDVs. So these vesicles are generated from mitochondria through a selective incorporation of protein cargos, which can be limited to the outer membrane or can include both outer, inner membrane, and matrix content. Ultrastructural analysis revealed that their size is between 70 to 150 nanometer in diameter, and that their scission does not require the fission machinery. Now, MDVs may interact with other intracellular compartments, such as multivesicular bodies, lysosomes, and peroxisomes, and they may also be incorporated into extracellular vesicles. Now, work from Heidi McBride's lab from 2008 was the first evidence showing direct interaction between mitochondria and peroxisomes by this mechanism. And since then, more work has been done in this field. Now, in order to understand the function of these NDVs in signaling and protein localization, it is essential to purify a large number of these vesicles and determine their cargo. And as you can imagine, this was not an easy task. And we had to direct our efforts in order to establish such system. So to do so, we decided to first examine whether mitochondria from Saccharomyces cerevisiae can generate such vesicles. And if so, we aim to analyze their content, their destination, and their function. So to this end, we established a platform in which we actually isolated mitochondria from yeast cells, resuspend them in isotonic buffer, and then ask whether these mitochondria release vesicles by using vesicles isolation methods. Now, the most exciting discovery, which is in the heart of our research and was published almost 2 years ago, was that yeast mitochondria release vesicles which are capable of generating an ATP via an active ATP synthase complex. Now, when we saw these results, we asked ourselves whether this ATP generation ability can be transferred to damaged mitochondria. And in order to address this question, we performed uptake experiments between respiratory deficient mitochondria, ATP2 knockout, and wild-type vesicles, and then we measured ATP by a luminescence assay. And as you all can see here, ATP levels of ATP2 knockout mitochondria were significantly increased after pre-incubating them with wild-type vesicles, which suggests that our MDVs can rejuvenate damaged mitochondria. So we got better understanding of one of the possible functions of our MDVs, but more questions are still open, and I would like to share with you some of our ongoing research. So for our first aim, it is well known that vesicles in general contain various cargos such as proteins, nucleic acids, and metabolites. And we, in our paper, were already able to determine our vesicles proteome. And now we are asking what other types of cargos are packed in our vesicles. And as you will see in a minute, we are focusing mainly on RNA. And for our second aim, as already showed you and told you, one of our major findings was that MDVs can rejuvenate damaged mitochondria, and we are asking whether this ability can be transferred to other organelles and cells. And hopefully, answering these questions, will provide crucial insights into the characteristic of MDVs and their potential for dealing with certain human diseases and therapies. So let's start with our first time, which is carried out by both. Now it is well known that RNA molecules are sorted into extracellular vesicles, influencing gene expression and immune responses. Now in order to understand whether our MDVs selectively carry RNA, we isolated RNA from both mitochondria and vesicles and generated an RNA-seq library. Now these methods enable us to ask which RNA sequences are present and if they are protein coding. So as you all can see here, PCA plotting of our RNA-seq library revealed two clearly separated clusters, one corresponding to mitochondria and the other to MVVs, indicating that our RNA populations are both reproducible and specific. Now, moreover, we validated the mitochondrial origin of our RNA-seq library by analyzing ribosomal RNA composition. And as you all can see here, both our populations, mitochondria and MVVs, showed low percentage of cytosolic RNA contamination, again supporting the purity of our RNA isolates. Now, based on our mass spec results, which we already published, we were able to detect some mitochondrial ribosomal proteins in our MDVs. Now, based on our new RNA-seq data, there is a hint that some tRNA molecules are also present in our MDVs, and we were able. 1 minute? Oh my God. Okay. Give me
You can have two, but no questions.
Okay. Okay, and there is a hint that some tRNA molecules are also present, and we could clearly detect all 8 mitochondrial encoded mRNAs inside our vesicles. And taken together, these results might suggest that an active translation may occur in our vesicles, and Bori is trying to reveal this mystery now by an in vitro translation system. Okay, moving forward, 2 minutes, like, are you sure it's 2 minutes? Are you sure?
The timer doesn't lie.
Okay. Okay, so I already showed you that our MDVs can rejuvenate yeast damaged mitochondria. We asked whether they can rejuvenate human mitochondria, and I will make it shortly. As you can see here, they did it. So these results suggest that the function of our MDVs may not be limited to yeast, and that MDVs are not necessarily confined to addressing ATP synthase deficiency alone, because here we worked on complex 1 deficient mitochondria, okay, isolated from human fibroblasts. Now, amazed by these results, we asked ourselves whether this complementation ability can also take place with higher eukaryote systems. So this time we performed uptake experiments between complex 1 deficient mitochondria and human MDVs. We isolated MDVs from human isolated mitochondria. And again, as you can see here, we got complementation. I'm making it short, I'm sorry. So these results open new opportunities for treating various conditions, and they led us to our next question which is the last, whether MDVs can be taken up by whole cells. Okay? Until now, we checked it just on mitochondria. And to make a long story short, the question is yes. We can see GFP-labeled MDVs inside whole cells, and this opened new opportunities for research. We have several collaborations. One is with Dr. Ziv Rotvogel from Kaplan, which is working on coronal wound healing. And together with his group, we are asking whether our MDVs could show similar effect. He saw an effect with applying external mitochondria, and we are doing the same with vesicles. Okay, so I don't need to sum up. You can read it alone, and thanks. Okay. Yes.
Can the MDVs survive the bloodstream?
Yes. We have a collaboration in rats in our hospital, and yes, we are working on a model of diabetes. Yes.
So all other questions at the end?
Quick. Ribosomal proteins or whole ribosomes?
No, not the whole ribosome.
Free protein, ribosomal proteins are very rare. You need to assemble in the ribosome.
it's mitochondria and ribosomal protein. Yet we don't see all the subunits. That's the results.
You can continue chatting about it after the session. And I love to stress my students, but not to break them, not try to do it. So just one before the last student talk.
I knew it was on purpose, Maya.
Yeah.
So, hi all, I'm really excited to be here today and discuss with you about my project, which is about peroxisomal proteins and how they modulate cellular response to oxidative stress. So, I think that if I would ask most of you what is your favorite organelle in the cell, you would probably not tell me the peroxisome. And you're right, the peroxisome was less studied over the years compared to other organelles in the cell, such as the mitochondria or the nucleus, but recently more and more evidence become clear that peroxisomes are really important for the homeostasis of the cells in stress and even in disease. And so peroxisomes are known for two general main roles. The first one is the oxidation of fatty acids, and this process releases a lot of heat and ROS. And therefore, it is only natural that the second role that peroxisomes are known for is the metabolism of ROS and dealing with oxidative stress. But the relationship of peroxisomes and oxidative stress started a very long time ago, in 1966, when two scientists, Weber and Baldwin, isolated from the cell's organelles that were called back then the microbodies. And they found that in these microbodies there are oxidative molecules such as H2O2 and also catalase that can deal with the stress. And so they decided to rename these microbodies to peroxisomes. And so I think that you can agree with me that oxidative stress is actually part of the peroxisome's identity. And from this day on, many great studies have been published about what is the relationship between peroxisomes and oxidative stress. So for example, it was shown that catalase can get out of peroxisomes to help the cell to cope with the stress. Later, it was shown that the glutathione is also important for the redox homeostasis of peroxisomes. And only recently it was shown that peroxisome can deal not only with their own ROS, but can also deal even with mitochondrial ROS. But although so many years of research has been conducted on peroxisomes and oxidative stress, we still don't know if there are mechanisms by which peroxisomes can regulate actually cellular recovery from oxidative stress. And so I decided to do something about it and go out to a journey. And in this journey I had a very specific aim, and this was to understand what is the active regulatory role of peroxisomal proteins in cellular response to oxidative stress. And so I thought that if I want to be successful, I need some kind of strategy. And if I want to look for a novel stress response, then I need to understand how a stress response typically looks like in the cell. So many times, not always, but a typical stress response starts with a stress. Then a sensor can sense this stress. A cascade of events is happening until eventually a transcription modulator can enter the nucleus to yield a transcriptional response, and then this yields adaptation and relief of the stress. Now, I wanted to look for a novel stress response cascade. And so I thought that the easiest for me would be to start with looking for the transcription modulator first. And this is because I can simply look for a protein that enters the nucleus under oxidative stress, and maybe this protein would be my transcription modulator. But I wasn't interested in any protein, I was interested only in peroxisomal proteins. And therefore what I did is take a collection of many proteins that are tagged either in their C or N terminus with a fluorophore, screen them under H2O2 conditions, and look for ones that are entering the nucleus under oxidative stress. All of these proteins are proteins that are somehow related to peroxisomes. And so one protein that I find that really strikingly enters the nucleus during H2O2 stress is PEX9. PEX9 is a peroxisomal protein that's known for being importing a subset of peroxisomal proteins to the matrix in different conditions, and it was actually discovered in our lab. And so it was not known that PEX9 enters the nucleus in any condition. It was not known that it has to do something with oxidative stress. So this was really exciting to know that not it only enters the nucleus, but it happens really, really rapidly. Even after 5 minutes, I already see it in the nucleus. So great, we have kind of a transcription modulator, but then we ask, could it be that it yields some kind of a transcriptional response, right? So to answer this, what I did is take yeast strains, then send them to transcriptomics when they were treated and with H2O2, and untreated, and look at the differential expression between the conditions. But I was interested to look for genes that are regulated specifically in these conditions only by PEX9 and not by the canonical stress response pathway for oxidative stress in yeast, which is modulated by ACK1 and SCN7. And so when you look at this heat map, you can see the list of genes that were regulated exclusively by PEX9 in this condition, and you can see that one of them is a really, really important one, which is TOR2. And TOR2 actually is an essential protein in yeast, in Saccharomyces cerevisiae. It is part of the TOR complexes, TOR complex 1 and TOR complex 2, which are master regulators of cellular growth and metabolism. So after seeing that PEX9 might downregulate TOR2 in the RNA level, we wanted to know what happens in the protein level. But it was very complicated to tag TOR2, and so we had to use CRISPR and tag it in between domains. And this was done by Mir Karmi from Nama Balkai lab. And I think that you can see here that in the control strain, treating the cells with H2O2 leads to changes in cellular localization of TOR2. And we think that it goes to the vacuole, therefore it might be downregulating it not only in the RNA level, but also in the protein level. And when we delete PEX9, that we don't see this effect anymore of H2O2 on TOR2. So great, we have a transcription modulator, and we might know what it does in the nucleus, right? But then, can somebody sense this stress and send it to the nucleus? So to answer who is the sensor, then I had no idea, so I needed to go over the whole genome. And to do that, I used a yeast library that in each and every strain, I have a different gene that is deleted or knocked down if it is an essential gene, then crossed it together with a strain that PEX9 is tagged with a fluorophore, and then looked under the microscope in H2O2 conditions. Now, if a certain gene that I'm deleted, then prevents the entrance of PEX9 to the nucleus. And what we found was very, very exciting, and this is that AVO1, which is an essential subunit of the TOR complex 2, modulates the entrance of PEX9 to the nucleus. And so what we think is that under H2O2 conditions, AVO1 can sense the oxidative stress, then modulate the entrance of PEX9 to the nucleus, and in the nucleus, PEX9 downregulates TOR2 that actually downregulates the whole activity of the TOR complex 2. Now, finally, we know that TOR complex 2 is very important for the cellular envelope homeostasis. It is also sitting on the plasma membrane. So we thought, what if AVO1 does not sense the H2O2 per se, but it actually senses the cellular envelope stress? And to answer this question, what we did is pre-treated the cells with sorbitol to ease a little bit the plasma membrane pressure. It is actually changing the osmolarity of the medium. And then treated with H2O2. And indeed, what we saw is that pre-treating the cells with sorbitol leads to less nuclear localization of PEX9 under H2O2 conditions. And so what we think that happens is that H2O2 leads to cellular envelope stress. This is known. And then AVO1 can sense this stress and modulates the entrance of PEX9 to the nucleus, which in turn downregulates the whole TOR2 complex. And with that, I want to thank all of you for listening. I want to thank Maya, the best mentor. I'm super lucky to be in this lab. To all the lab members, specifically to Yeist, Rosario and Olga for helping me with this project. To Amir and Iqbal from the NCPM, the Weizmann, and to Miri from the Nama Balkai lab. And one more thing before questions. Okay, so Noga from the lab and I are organizing a conference at the Weizmann Institute about stress. Okay, so we will be really, really, really happy to see you there, and it is super exciting. So save the date, May 6th, and I'm happy to take questions if I
Conference details if anybody wants to take a picture. Perfectly, we have time for one question. And now you tell me something raising their hand?
No, I guess it was still going.
No, it's amazing. I can't ask a question because it's my project. I'm sure you have the best question.
No guideline.
תודה רבה, מאיה.
No, I just wanted to say, I have a feeling this is going on in Albacans as well, and we should talk.
Ah, good. Okay.
Okay, cool. Thank you.
Last but not least, exactly, Kobe Simpson-Lavie from Martin Kupiek's lab. Thank you, Kobe, for finishing off this great
Thank you. So I'm going to talk about how some of my work I'm doing at the moment on how the SNF1 protein kinase regulates the low iron stress response in Escherichia. So in yeast, we've talked lots about yeast, but their main use in history is, of course, in fermentation, which is the conversion of the sugars glucose, fructose, and mannose to ethanol, and we use that to make our bread, our wine, our beer. And other carbon sources, such as sucrose, galactose, glycerol, ethanol, oleate, and so on, actually every other carbon source, is respired to carbon dioxide and water and lots of ATP. Now, if you have glucose, fructose, or mannose in the media with any other sugar, it will suppress respiration. This is called catabolite repression. And this is achieved because these fermentative sugars turn off the SNF1 kinase complex, which is comprised, oh no, hang on, sorry, which is, which one's the laser? This one? Okay, great. Which is comprised of the SNF1 kinase itself, a beta protein, and a gamma protein. And the SNF1 complex is equal to the mammalian AMPK, and this complex promotes respiration both at the transcriptional level and at the level of direct regulation of proteins by phosphorylating them. So the SNF1 kinase has several domains, but we'll have to quickly talk about them. We have a kinase domain at the N-terminal, and there's a phosphorylation site at T210, which is phosphorylated when SNF1 is active in the absence of glucose. In the C-terminal half, there's an autoinhibition motif, which is blocked when SNF1 is active by the gamma subunit, and one of three beta subunits binds to the far terminus. This region also has a stimulation site at lysine 549, which is stimulated when in the presence of glucose to turn off SNF1. Finally, at the far end terminus in the pre-kinase region, there is a polyhis motif of 13 histidines in a row, a glycine and one more histidine, which is going to be the focus of this talk today. And this region is conserved amongst the yeast, but not amongst filamentous fungi, and not in high eukaryotes. So how is SNF1 regulating processes? So this is from a paper published a couple of years ago. So in presence of glucose, the SNF1 complex is dissociated, and its target genes are not transcribed. Now, if say add in a new fact into this with iron, so when iron is present in the extracellular media, it is transported into the cell by pumps such as pumps by FET3. It is sent from mitochondria and converted into iron sulfur clusters. These then bind and restrain the AFT1 transcription factor from entering the nucleus, thus preventing expression of target genes such as FET3. If we now remove the glucose, SNF1 translocates into the nucleus with its gamma unit and the beta subunit. It's phosphorylated. The pre-kinase region with histidines is protonated, and this is now active and turns on expression of genes such as AGH2. And now we're going to remove the iron. Go down here. To remove the iron, this then means less iron sulfur clusters present. AFT1 is now released and enters the nucleus, where it's going to do two things. First of all, it is going to bind to SNF1 at the protonated polyhistine sequence and inhibit it, thus preventing expression of AGH2. And it also turns on its own target genes such as FET3 and CCH2, which is very important. We aim here to scavenge as much iron as possible from extracellular media, and also to reduce, basically to conserve the iron to divert it away from processes such as respiration and towards enzymes which are more important, such as for DNA repair and transcription. So this is what I published already. And now I turn to a related question. Okay, we've seen now in these past data that AFT1 inhibits SNF1 activity. Now we're going to look at the other way around and see if SNF1 inhibits AFT1, inhibits FET3 expression, for example. So I looked at FET3 expression in different carbon sources. We see very clearly that you need glucose to get a FET3 expression. It is not expressed in other carbon sources at all. But very surprisingly, very surprisingly, we see an increase in expression in the absence of SNF1, even in glucose-grown cells, where SNF1 is inactive anyway. So I checked the interaction between AFT1 and SNF1, and the two proteins interact very strongly in ethanol, but even in glucose, there's a 20% expression, which is further lowered when the polyhis is eliminated, so comparing here to here. So there's an interaction, and it is dependent on this polyhis motif. Now check mutations in SNF1, see if this can affect FET3 signaling. We do this by, we grow the cells in glucose, all these experiments are in glucose now, and we add an ion chelator called BPS, and that locks up the ion, generating a low ion response. So as we already said, in the SNF1 knockout, there is higher expression of FET3. Surprisingly, in a kinase dead mutant of SNF1, the K84R, there is no change at all. When we eliminate the polyhis, either by total deletion or by substitution of the histines to alanine, we get this higher expression of FET3. So this tells us that the polyhis is important, but kinase activity itself is not actually important at all. It's not about kinase activity. And remember, we're still in glucose all the time here. So indeed, if we take the first 53 amino acids of SNF1, which contains this polyhis motif, and just overexpress it, and we have two ways to overexpress it. One is on its own, and one also fused to a NOS. So we overexpress it and monitor the effect of adding FET3 upon the effect of adding BPS on FET3 expression. And we see that when first of all we have a nuclear version, there is no effect at all, doesn't do anything at all. But when we have a cytoplasmic version of the normal amino acids 1 to 53, no mutations, we see a big decrease in FET3 expression. And again, this is totally dependent on the polyhis, because if we eliminate the polyhis motif, we have no effect at all. So if this all being the case, we don't need kinase activity. How could this work? One possibility is that it is due to SNF1 abundance. And we see here that SNF1 abundance increases when cells are transferred from glucose to ethanol. So we have a way we can actually make SNF1 levels change. So bearing this in mind, we now do an experiment where we're going to grow the cells overnight in glucose, then change the cells to keep in glucose or switch to ethanol to cause accumulation of SNF1, as we saw. Now we have to add glucose because we know we don't get FET3 expression when we only get expression in glucose, and add BPS. And we see here a reduction of FET3 expression in the cells that were previously grown in ethanol. And again, this is totally dependent on SNF1, no change here, and also on the polyhis, because there's no change here. And if we take a mutant of SNF1 which has high abundance due to lack of assimilation, we see here it is low, but also no change. So the effect, this effect we're seeing is due to SNF1 abundance and not activity. So to conclude, the SNF1 protein inhibits FET3 expression in glucose media, even in the absence of kinase activity. The N-terminal polyhis sequence of SNF1 interacts with AFT1 and is required for inhibition of FET3 expression. SNF1 protein levels increase in the absence of glucose. prior growth of cells, the absence of glucose diminishes AFT1 activity, and this moderates the inhibition of respiration processes by the low iron response. So thank you very much, and I have questions for you. Which protein which I've talked about is represented by these pictures? It's fat-free, they're pumping iron. Okay, any actual questions? Yes.
Do you think that the histidine enrichment motif may be sensing the lack of iron?
Oh, that was my first thought, but I found that the histine motif is binding to AFT1, so it's an indirect sense. It's indirectly working on this process.
So would it be binding because there is less iron, so it's free to bind to AFT1?
No, AFT in the absence of iron, AFT1 will go to the nucleus where it will encounter SNF1. So it's, yeah, that was my original idea. Yes, it'd be really cool, histine coordinating on the iron, but unfortunately not.
So thanks to all of you for sticking with us all today. We're coming to the end of this fantastic celebration of stress and science. You know that the secret for having a happy life and not being stressed is to every day take a couple of minutes to remember and be grateful for the things that you do have. So I would like to end by telling you a couple of things that I'm grateful for. First of all, I'm grateful to Martin. I think Martin is the person that created the Yeast Club, right? Many years ago.
I don't remember anymore.
Yes. So Martin, I'm really grateful to you for starting this amazing tradition. I'm also really... We're all grateful. Yes, we're all grateful to you. And let's I'm really grateful to Shai that agreed to take on the next phase.
That's the way it works.
Linkage. I'm really grateful to the Institute for Advanced Study staff that worked tirelessly to enable this day to go on and organize everything around it, so they really deserve a round of applause. They're not here, but we can. I'm super grateful to all of our speakers and panel members for sharing your science, your insights, for sticking time. Everyone except Amir. And it was really fantastic to hear the amazing diversity of research done on yeast, from basic cell biology to kind of technology and therapeutics, and it's great to see how versatile this organism is. I'm grateful to all of you, the audience, that you were so actively participating, asking questions. It's bringing with you this environment of friendship and curiosity and community and love and excitement for science. And it's not trivial to take a day from everybody's very, very busy life and come and spend it together. So I think this, to me, this means a lot. So I'm really grateful to all of you. But most of all, I'm grateful, you know it's coming, Ayala. But I have to tell you one thing. There are some people that are hard to work with. There are some people that are nice to work with. But there are very few people like Ayala. Every time I talk to you, you make my heart grow. You're an amazing, amazing friend, an amazing person to have great ideas with. So it's really been a delight.
Can I add that we all needed this?
As a
This
test.
was your very, very important. Thank you, Maya. And Maya.